Hello and good afternoon. My name is Rajiv Nepal. Today I'll be talking about the breadth of coverage of meningococcal B factor binding protein vaccine, MenB-fHbp. This is our disclosure slide. In the case of meningococcal B bacteria, the polysaccharide capsule could not be used as a vaccine antigen because of its structural similarity to human neural tissue. Hence, surface expressed proteins were used as antigens to develop vaccines against MenB. Since proteins on MenB strains have high degree of genetic heterogeneity, for a protein-based MenB vaccine to be broadly protective against circulating strains, the antibodies generated need to protect against most of those variants. The high diversity of MenB strains paired with the rapidity of invasive meningococcal disease creates several issues. For public health, it may limit the ability to ascertain vaccine coverage of a particular strain in real time during public health emergencies. Similarly, healthcare practitioners vaccinating individuals prophylactically would like to understand the potential coverage that a MenB vaccine would have against circulating strains. In both scenarios, a MenB vaccine with coverage against most of the circulating strains would be ideal. Factor binding protein, FHBP, emerged as an ideal meningococcal antigen for broad coverage. So let's discuss the rationale first. As I indicated earlier, there are many different variants of FHBP. Based on genetic and immunological basis, they can be classified into two distinct subfamilies, A and B. Studies have demonstrated that lipidated factor binding protein antigens are more immunogenic than their non-lipidated counterparts, and they elicit broadly cross-reactive responses, usually to variants within the same subfamily but not to other subfamily. Hence, in order to provide broad coverage of MenB strains, MenB-fHbp vaccine contains one lipidated FHbp variant from each of the two subfamilies A and B. So in this slide, by using serum bactericidal assay, SBA, which measures the killing of meningococci in the presence of vaccine-induced antibodies, we show how MenB-fHbp-induced antibodies can kill diverse strains carrying FHbp variants from either subfamily A or B. So the two subfamilies are represented here as two separate flowers. The vaccine antigens A05 and B01 are represented in the center of each flower. The approach to evaluating breadth of coverage started with SBA assays showing the killing of 4 primary strains, 2 with subfamily A and 2 with subfamily B FHbP. To further assess the breadth of coverage, 10 additional test strains from both subfamilies were tested and shown to be killed in SBA. A number of additional studies have demonstrated the killing of several meningococcal strains with diverse FHbP variants as shown here. Interestingly, 9 out of 10 most prevalent Canadian FHbP variants from MenB strains that have caused invasive diseases here in Canada, they have already been tested and shown to be killed by MenB-fHbp induced antibodies. So one thing about killing of MenB in SBA is that the surface expression of FHbP is very important. Sufficient expression is required for killing, and isolate with very low FHbP expression on the surface may not be susceptible to killing in SBA. So in the next step, and in the interest of time, I'm not showing the data here, what we did was we established a predictive threshold of FHbP expression where most of those strains were susceptible in SBA. So once we had that threshold, then we entered into another stage. So finally here, Beringer et al. In their 2020 publication assessed the expression of FHbP among Canadian meningococcal B isolates, which were collected as part of the Canada's impact surveillance between 2010 and 2012. 91% of those isolates expressed FHbP at levels above the threshold that I just described and are predicted to be killed. So in conclusion, and as a take-home message, 9 out of 10 Canadian MenB strains that caused invasive diseases here in Canada were predicted to be covered by antibodies generated by the MenB-fHbp vaccine. So with that, I would like to conclude my presentation. I'll be more than happy to answer if you have any questions, so I'll stick around until the end of the session. Thank you very much for your attention.